Today we're going to conclude our series of lectures with an examination of cancer and the biology of human cancer specifically. This is one of the most important public health issues in America today. There are more than 6 to 7 million new cases of cancer worldwide per year, and more than 1 million cases in the United States if you exclude the nearly million non-melanoma skin cancers. There are about 8 million Americans alive today who have had cancer, and of those, about 5 million are considered cured. Now, this is the second leading cause of death in America behind heart disease. And as we get better with our treatment and prevention of heart disease, this may soon become the leading cause of death. Cancer is a genetic disease. And I say that right up front because you remember we had some very specific definitions about what is a genetic disease. It's a disease in the genes, a disease in the DNA. It is not necessarily inheritable or congenital. In fact, it is rare for cancer to be either inherited or congenital. Less than 5% of all the cancers fall into that category. But it is a genetic damage that occurs in our somatic cells more often rather than in our germ cells. If we look at the nature of DNA, DNA is what we would call a mutable or mutatable molecule. If DNA were forged in steel as a molecule and could not be changed, there would be no chance of mutations or errors. But it would also mean that we would still be living in the primordial soup of eons ago because there would be no possibility for evolution. The fact is that DNA is a very fragile molecule that's easily damaged and easily changed. Mutation is the word for change in the DNA, structural change. And because of that, we were able to evolve. The evolution with natural selection could occur and got us here to where we are today, but with it brought along the other side of this sword, and that is we are able to get cancer, because cancer results directly from changes in the DNA. Now, again, I'm almost always going to be talking about the somatic DNA, our body cells. Cancer should be looked at as a microevolutionary process at its very basic level. The evolution that's going on is taking place at a molecular level, and there is natural selection of the fittest gene. And in this case, some of these genes are going to be very bad for us. Just as evolution is natural selection of the fittest genes in normal processes, so it is for letting cancer get out of control. Cancer is also, unfortunately, very largely a self-inflicted wound in our societies today. 80% or more of all the cancers are probably environmentally caused. Chemicals, especially agriculture and industrial chemicals, are a leading cause of mutations in the somatic cells of our bodies and a leading cause of many, many kinds of cancers. Ionizing radiation is another source of injury to our DNA. This is a physical carcinogen. And because of lifestyle changes and changes in our atmosphere, such as the hole in the ozone layer, we've exposed ourselves to more and more ionizing radiation, specifically what we call UVB light. Asbestos and radon are two more physical carcinogens that we have brought upon ourselves by disturbing our environment. And finally, there are viruses, which are sexually transmitted diseases, which also go along with some types of cancer. So basically, these environmental carcinogens, or substances that cause cancer, are ones that we can find and prevent if we could prevent our exposure. In some cases, we're just not willing to do it. For example, cigarette smoking, very easy cause and effect, and yet people know this. Almost anybody who can read or write knows that cigarette smoking causes cancer. Yet very few people are willing to stop the cigarette smoking behavior in order to prevent the cancer. So just knowing what causes them may not be entirely adequate to get people to change what's going on in our world. Same with the use of insecticides, pesticides, and industrial chemicals, major cause of disability and cancer in people worldwide, yet we continue to use them in places where they may or may not be necessary. Let's define cancer first and take a look at the characteristics that are common to all cancers. Now, this is not necessarily a definition you'd find in a dictionary, but I think it works very, very well. And if you apply this to almost anything you are considering as a candidate for a cancer, this will apply and work every time. The first thing that happens is cancer is a failure of the stem cells to differentiate. Cancer does not start in somatic mature cells, fully differentiated cells. We used to think that it did. We had something called the histogenic assumption, which said that, for example, the cell in the duct of a breast that is fully matured and functioning gets injured somehow or given a signal and loses differentiation. We talked of loss of differentiation and then becomes cancer. That's totally incorrect. Textbooks even today use the term loss of differentiation. The cell cannot lose differentiation. A fully matured cell can only be that cell or die. It can't become a cancer cell. And there's nothing we can do to make it that. Most cells are pretty much finished with their replication anyway at that point. The average cell only replicates 30 to 50 times in a lifetime before it stops replicating and becomes senescent. It's one of the reasons we age. Cancer cells, on the other hand, come from stem cells of various organs that are determined, for example, stem cells in the colon that are going to replace the dead cells of the colon later, stem cells in our intestine, our skin, our respiratory tract, different places where those cells are now on a signal going to emerge and should differentiate, but become injured and accumulate mutations which don't allow it to happen. So they fail to differentiate rather than losing differentiation. This should immediately bring up a question a sixth grade class also asked me. They said, I had told them about being born with their full complement of brain cells, and the kids raised their hands and said, well, then how do you get brain cancer? If there are no stem cells, or very few, to rise up and try to differentiate, how can a brain cell fail to differentiate? And they were quite correct. We don't get brain cancer. We don't get cancer from the neurons of our brain cells. We get them, as I mentioned earlier, from the supporting cells, the glial cells that hold the brain together. They're called astrocytes and other kinds of cells, gliomas. These cells are turning over. Those support cells are turning over, and that's where brain cancer occurs. So it backs up what we're thinking about failure to differentiate. So that's number one in our chart here, to define cancer. If we look at the next one, is the potential to invade. Normal benign cells, and you're going to hear me say benign and malignant. These are two gross categories. Benign means bringer of good, malignant means bringer of bad or evil. And benign cells are cells which know their place and do not leave their assigned position. There is in many tissues something called a basement membrane, and those benign cells will remain behind the basement membrane and not get out into other tissues. Cancer cells have the potential to invade. Potential is a very important word here because they don't have to invade yet to be called or diagnosed as cancer. I'm going to show you what that means. There are benign tumors. There are growths of cells. Tumor is just an enlargement of a mass. It doesn't mean cancer. And there are cells that grow abnormally large in mass, but a benign one will never, by definition, break through its basement membrane. It will never escape into other tissues. Now, cancer also is a generic term. Cancer refers to all malignancies. You'll hear me later talk about carcinomas and sarcomas, and we'll get to that in a minute. That's number two, the potential to invade. Remember that invasion does not have to take place for this to be called cancer. So let's look at a slide and try to illustrate that. One of the most common cancers is cancer of the breast, most common cancer among women. This is the picture you've seen of the breast, and we're going to take a slice right there through one of the ducts. Cancers arising in the breast ducts comprise about 80% of all breast cancers. Those that arise in the lobule comprise about 19%, and the rest about 1% for the different structures of the breast. However, the most common here will serve as an excellent example all the way through our discussion. If we take that cross-section and we look at the next picture, the normal duct on section looks like this. There is an outer basement membrane which completely encircles the duct. There is a lining of epithelial cells. Since these are in connection with the outside through the nipple, they are epithelial cells. And they secrete either a clear fluid, which fills up the lumen of the cell, or they may secrete milk during lactation. These cells are usually very orderly arranged, just like this, sometimes 2 layers, but rarely any more. Now, these normal cells cannot get out into the tissues. They cannot penetrate the basement membrane. If we have a situation where the cells begin to replicate rapidly, they can be benign cells and grow in here and fill the lumen, or they can become duct cancer cells. This is called DCIS, duct carcinoma in situ. Carcinoma refers to cancer in epithelial cells. So the lining of your intestine or breast ducts are epithelial cells. That's why breast cancer is a carcinoma. This is a very common disorder. What's happening here is that these cells are replicating very rapidly without constraint and filling up the whole center of the duct. These cells have not broken through to the outside, but they're fully capable of it. If we take a biopsy at this time, the pathologist can make the diagnosis of cancer because this cell, we know, has the potential to invade at some point. It just hasn't done it yet. So it still fulfills this category of potential to invade. Now, what happens when this potential is expressed is the cell has the ability to break through the basement membrane and, using various motility techniques, move on out into the surrounding tissue of the breast and begin to grow there. That's called local invasion. So whether it has invaded or has potentially invaded, this ability in its genome, the proteins and chemicals that make it possible, give it the potential, and it's part of my definition. If we move on to the next criterion, it has the potential to metastasize. Metastasis means growth at another place. Again, look at the word potential. What happens in metastases is that after the invasion takes place and there is growth in the local area, this is local invasion, the cancer cell can also enter an artery, a vein, or a lymphatic channel, get a free ride to somewhere else in the body, and then arrive at another capillary system which may trap it or find a very fertile ground. For example, breast cancer cells arriving in the ovary find lots of estrogen, which they like, and it may stay there and grow. The cancer cells of whatever kind will then go off to a distant organ and begin to grow here, and this is a representation of clumps of metastases, singular as metastasis, of cancer cells. Now this brings us to a definition. We talk about primary and metastatic disease. You often will hear a person in this country say, I had an aunt who died of liver cancer. That would be highly unusual. Primary liver cancer is very, very rare in America. It's more common worldwide, but not here. What that patient's aunt really had in a particular case is a primary breast cancer that started in the ducts, locally invaded, metastasized, and went to the liver, where it grew and killed the patient. That is not liver cancer. That's still breast cancer, primary in the breast, metastatic to the liver. a common place for breast cancers to spread, as well as lung. Now, if I went to the pathologist with a piece I took out of a liver, I said, I had a patient who seems to have a lump in the liver, and I biopsied it, and I take that piece to the pathologist, or in this case, don't say where I got it. I say, here, take a look at that. What's that? That pathologist will be able to say, oh, that's breast cancer. And he said, where'd you get it? Well, in that case, I didn't get it out of the breast, I got it out of the liver. It's still breast cancer. It doesn't change. The primary never changes. The metastasis is merely another growth site. So it's important to remember that you want to know the primary, because you would treat a primary liver cancer completely differently from treating a breast cancer that had spread to the liver. Remember all those lymphatics and veins that came out of the digestive tract, especially the colon, and ran up into the portal circulation in the liver. That's why so many cancers of the colon end up in the liver. They go through and invade those veins, circulate to the next small capillary bed, which happens to be the liver. Very common source of metastatic disease is the liver. Finally, if we go back now and look at my fourth criterion, there's a designed lethality in cancer. Benign tumors, by contrast, don't kill the patients. They can occasionally kill by accident. There is a tumor of the covering of the brain, remember the dura. There's something called a meningioma. Oma means a benign form of tumor on the dura that grows inside the skull. Now, it can't invade. It can't metastasize, but it's in a closed case. So if it grows, it's going to kill the patient by pressure on the brain, even though by nature it was benign, and this is an accident. There's something called a myxoma, a fatty tumor in the heart that flops around attached to the wall. It can get big enough to accidentally get into a valve, plug it off, and kill the patient. But it was a benign tumor. It can't invade. It can't metastasize. It has no potential. It just kills the patient by accident. In cancer, there's a designed-in lethality. These cancers will eventually, some quick, some later, invade, grow locally, metastasize, and get to a vital organ. While the cancer's in the breast, it cannot kill the patient. The breast is not a vital organ. It does not support vital function except for the fetus. So the woman could not be killed by the breast cancer in her breast. It's got to get to a vital organ, which in the case of most cancers is either brain, bone, because it destroys the bone marrow, where our immune system is, lung and liver. Those are the major areas and the causes of death in most cancers. So we have these four criteria: failure to differentiate, potential to invade, potential to metastasize, and designed in lethality. Now, in looking at this and now understanding the difference between benign and malignant and primary and metastatic disease, we may want to look at some of the terms. The ending OMA, which means a tumor or a mass, is almost always signifying a benign state. There are two exceptions, and doctors have not corrected them. We always talk this way, so you just need to understand. Melanoma is a malignant tumor of the skin. It has dark pigment. It should be a melanocarcinoma. It's never called that. It's a melanoma, and although it is malignant, it has the ending that implies it's benign. You just have to accept that. The other one is lymphoma. Lymphoid cancer should be lymphosarcoma. I'll tell you why in a second. It is not. It's always called a lymphoma. There's no such thing as a benign lymphoma. They're all malignant. There's lymphoid hyperplasia. That would be a benign form. But we don't use this term to refer to the malignancy. Now, there's basically two kinds of terms we use. The carcinomas refer to any epithelial tissue or tissue on the outside. The lining of your intestine really is on the outside. It's just the inner part of the tube. So epithelial cells, like in the skin or linings of organs, the breast duct, are all carcinomas. Colon carcinoma, breast carcinoma, melanocarcinoma is what it should be, but it's not. Squamous cell carcinoma of the esophagus, squamous or basal cell carcinomas of the skin. They're all epithelial tumors. Sarcomas refer to what we call mesothelial tumors. That first series of tissues I showed you in the embryologic stages, that's in the middle. Connective tissue, bones, muscle. And then we name them. For example, skeletal muscle has the name rhabdomyo. If it's benign, we would call it a rhabdomyoma. If it's malignant, we would call it a rhabdomyosarcoma. Smooth muscle is called lyo. So we have a leiomyoma, which is a benign tumor in the uterus. We have a leiomyosarcoma. That's when it's malignant. Same thing with other tumors of the epithelial cells. We can have an adenoma. You saw that there were adrenal cortical adenomas. It's a gland. Adenoma means gland. So gland tumors that secrete things can have a benign form, adenomas, or you can get an adenocarcinoma. Now, the breast is an adenocarcinoma because it's a gland. We describe adenocarcinoma of the breast, and then a subdivision, ductal or lobular, invasive. That would be the way we'd categorize it. Going to hear these terms a lot. So what happens to make something a carcinogen? To make something a cancer, how do these cells get injured? There are basically 3 different categories of carcinogens in our environment, and they can injure our somatic cells. The first and perhaps the biggest group are chemical carcinogens. These include vinyl chloride, all kinds of insecticides and pesticides that you get on your food in agriculture, industrial chemicals, a whole wide range, literally thousands of these chemicals, if they get into our cells in sufficient quantity, can do something that prevents us from replicating our DNA properly. All that's happening in cancer is that that stem cell is now going to start dividing and trying to differentiate. It's going to peel apart its DNA, and it's going to want to line up a complementary or mirror image DNA so that both new cells will have the correct sequence and code in the entire genome of its DNA. Chemicals tend to form adducts. The chemical molecule gets on the half of the DNA that's been exposed and locks on and stays there. Some chemicals form more adducts than others. The more adducts, the more mutations. Now that mutation gets on there, and so when the DNA unzips and tries to replicate, there's a code that can't be read. Instead of A, G, G, C, T, 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 C, A, as we've seen in DNA, there's going to be a block that's illegible for the complementary strand of DNA, and it will be randomly wrong. If it occurs in an important gene or protein, and I'll tell you what those are, we have an error in the replication. And that's how almost all chemical carcinogens cause the errors in the DNA. They form an adduct, they block correct transcription of the DNA to make the new daughter cells, and one of those somatic cells will now have an error in it. When it replicates, it's going to repeat the error. The gibberish in there is now going to be copied by the next daughter cell, and we have a whole clone that's going to go down the line wrong. That's part of what happens in chemical processes of carcinogenesis. We also have biologic carcinogens, and these include primarily viruses and one bacterium. I'll mention the bacterium first. It's the Helicobacter pylori, and the way that works is by stimulating the rapid production of repair cells, we believe, in the stomach. It causes stomach cancer. It's an unusual one. It's the only bacteria right now that we know of that can cause cancer. The viruses are a different matter. There are a number of viruses which could make you say then, in some cases, cancer is an infectious disease, and in some cases, a sexually transmitted disease. Viruses come in 2 flavors. There are DNA viruses and a whole other group called RNA or retroviruses. DNA viruses include hepatitis B, human papillomavirus, which causes warts, venereal sexually transmitted warts. And these viruses are very, very common in the world. Hepatitis B virus is widespread, although we have a vaccine against it now. And human papillomavirus is almost ubiquitous. Sexually active people over the age of 20 or 23 almost all show antibodies to having at least at one time been infected by these viruses. The DNA viruses have a really interesting way of getting in and starting up the cancer process. Remember, viruses invade cells and they replicate inside the cell. The DNA virus has a gene that tells its machinery to replicate really, really fast, because viruses want to make a lot of viruses. The DNA virus gets in your cell. In hepatitis, it's a liver cell. In human papillomavirus, it's on the uterine cervix. Gets into that cell, starts to replicate, and the human body response may kill the virus and make it fail. But it's got a gene there that I'm going to tell you about in a little bit that says replicate like crazy. That gene gets left behind in a living cell, is incorporated into the genome, and now the cell has a gene that says replicate like crazy, which is what cancer cells do. It takes away the restraint of growth. We'll come back to that. The RNA viruses, like HIV, are different. RNA viruses have already visited a cell that has cancer. They successfully replicate, and they pick up that gene that the cancer cell had that says replicate fast like crazy and take it into their genome. They then move on and infect another cell, but they fail and they drop that gene off there. So the cancer gene was a hitchhiker with the RNA virus. That's basically the way we get carcinogenesis from viruses, and these are definitely infectious diseases in this case. Obviously, cancer formation from a chemical is not a transmissible disease. Finally, we have physical carcinogens, which are radiation, ultraviolet B light, are two of the most important. What these are energy disruptions. Basically, a photon of light comes down and strikes the DNA, because it can go right through the cell, causes a break or a disruption, or many of them within that cell, and that is your mutation. When that cell replicates, there's going to be an error because there's a break in the DNA, either one or both strands. Photon frequency happens to be the same distance physically as there is across the strands, so they match up perfectly to nail DNA. Asbestos is another very common carcinogen in our environment, which we have released into our environment, and asbestos works as a micro spear. It's as if you fired a spear into the cell. You disrupt the nuclear and cytoplasmic chemicals, and they are not meant to mix. They do mix, and we get abnormalities in reproduction. So those are the carcinogens that cause us to have cancer. Now, there are mechanisms of how does this actually work? Why does it really happen? How do we get cancer in the cell? And I'll show you that next. We have two things going on. In our genome, we have, in the normal genome, we have a gene or set of genes that regulate the rapidity of growth. When that gene signals the cell, which is very infrequently, the cell will replicate and make two cells. I told you in a normal cell this only happens 30 to 50 times in its lifetime. Now, then that cell becomes senescent and dies, we need a new cell. Remember, it doesn't happen even frequently enough for us to see the mitotic figures of division when we look at a slide in normal tissue, but we see many of them in cancer. So cancer is replicating really fast. That is called a proto-oncogene. Oncogene means a cancer gene. The proto-oncogene was discovered when we discovered the cancer genes and worked backwards and saw that the oncogene was really a damaged or mutated proto-oncogene, and that's how it got its name. So in our normal machinery, we have a gene that regulates the rate of growth. Now, when a mutation occurs, we might injure that region of the genome, of the DNA, and switch on the oncogene, the proto-oncogene, so it now doesn't turn off. It can signal the cell to replicate without rest at high speeds, and not take a break. Completely abnormal, and we now have an oncogene. This is the first step. Another thing is going to happen there. This is as if we got on the highway and we put our foot down on the throttle and we got the car up to 115 miles an hour and we took our foot off and nothing happened. The throttle was stuck. That's what happens when you get proto-oncogene activation to an oncogene by mutation. It's a random event, and that's why we don't get cancer more than we do. But when it does, we're now out on the highway and we're driving at 110 miles an hour and we can't get the accelerator to come on back. At the same time, in the same cell, we have another problem. We look here, these are called tumor suppressor genes. Tumor suppressor genes are natural components of every cell in the body. The most famous is called P53, and I'll tell you about P53. Tumor suppressor genes have the job of looking at every replicated strand of DNA. It actually doesn't look at the sequence, but it looks at the products, the proteins. And it says, yes, GGTA, this is the way it's supposed to be. And it comes to one segment and says, that's not the way it's supposed to be. This is wrong. The gene is called the guardian of the genome. P53 molecule or protein looks at the genome and says there's something wrong with this genome. Stop replication, don't go ahead. The P53 then signals a whole chain of other molecules and proteins, but the net effect is that replication is halted long enough for that error to be corrected. It can actually snip out DNA sequences that are wrong and put in the correct sequences. There's a major repair mechanism to prevent the cell from reproducing with inaccurate information. Very critical. Now, if the P53 mutates and becomes inactivated, then it can't tell the cell to stop. P53 can also say, that's too much damage, commit what's called apoptosis. And apoptosis is programmed cell death. It's not like getting stop of blood flow or stopping oxygen. It's actually a chemical suicide pill, just like the cyanide a spy carries in his tooth. There's a suicide pill in a cell, and P53 can trigger it, saying there's too much damage, it's too dangerous, just kill the cell and me along with it. That's cell suicide, programmed cell death, or apoptosis. If the P53 here, our tumor suppressor gene, among other tumor suppressor genes, is inactivated at the same time as you get activation of a proto-oncogene, then we have the car on the highway going 110. Our only choice is to put on the brakes, and in this case, the brakes go to the floor, and we have no brakes. We have disaster in the making. That is the analogy of what's happening in this cell. We've got rapid replication, we've got no brakes, we can't stop acceleration, the cell is out of control, and it's not listening to the other cells that are telling it what to do. Remember we talked about autocrine signals. This cell is now signaling itself to replicate very rapidly. Analogies are only good as far as they go, but I want to give you another one. If I gave you a typewriter with 4 keys, it had the 4 letters of the genetic code, and I said, go ahead, type this out at 60 letters per minute, and you'd get it right every time. If I said, I'd like that at 120 letters per minute, well, when we got up to 500 letters per minute, you would definitely start making errors. The same thing happens in DNA replication. The more time that you have to make corrections, the better the replication will be. The literal physical time shortening and rapid replication will make more errors. What's going to happen? We're going to have more mutations. We're going to activate more oncogenes. We're going to inactivate more tumor suppressor genes. Now, what happens if the mutation is in that huge silent area of the genome? Nothing. Doesn't matter. What happens if the error is so huge that the whole replication process collapses and fails? That doesn't matter. That cell will just die and another cell will take its place. It's that little fine place in the genome where either oncogenes or proto-oncogenes are regulating the rate of growth, or tumor suppressor genes are being made where we activate one, inactivate the other, that we've got the recipe to make more errors and accumulate more mutations. And again, if they're lethal mutations, doesn't matter. But a certain percent of these will come out making the cell turn over too fast. And those cells are what we call cancer. Along with those processes come attributes. If we go back now to this slide and look at our duct cell, what are the mechanisms? I've told you what happens when these processes go on. And this, by the way, is called initiation. This first process of inactivation of the tumor suppressor cells, activation of the oncogenes, is called initiation. That's an initiated cell. And it happens by chemicals, DNA, viruses, bacteria, and physical injuries. The next step is a process called promotion. We need to whip those cells that are able to replicate fast. And in breast cancer, one of the promoters is estrogen. You can't get a breast cancer from estrogen alone. You need the initiated cell. Once the cell is initiated by mutation, then the estrogen whips it along. It says, replicate as fast as you can, which it does. It is still not yet cancer. At this point, it could reverse if you don't get enough promotion or you don't get enough replication and you don't accumulate some more mutations. It will cross a line called conversion when it has all the mutations it needs to do everything I pointed to before, the 4 processes. And then once it crosses the line, there's no going back. It is cancer. It's irreversible. It's a cancer cell. And then the 4th stage is progression, where it goes from microscopic to big, and that's clinical cancer. The time frame from initiation in most solid tumors of the body to the time we have a clinical cancer and progression, the cancer that patient that walks into my office with something I can diagnose and treat, is anywhere from 10 to 40 years. It's a very long period. Why? Because it has to accumulate a perfect set of mutations. The fastest cancers we know about, or the fastest malignancy, occurred after Hiroshima. We irradiated hundreds and hundreds of people who survived, thousands of them. We killed 100,000 who didn't survive. And the first cancer that appeared in the survivors was leukemia in children. Still took 7 years. So this is a long process. What we want to be doing as surgeons is, I don't want to be out here on this side in the place where progression is going on, working on this big problem. I'd really be back way over here at initiation and stopping it from happening. The medical profession wants to be and intervene back here and prevent. Or we want to diagnose this problem very early in the next stage, which we can't even find now, and do something about it. And we surgeons are the dinosaurs who are over there basically cutting it out. That's as crude as you get in terms of a savage way to deal with this kind of problem. How does this actually work on a more basic level? What is invasion and metastasis? Well, if you go back to this picture, these cancer cells have actually developed mechanisms for doing all the things we said were required in our 4 definitions. The first thing they have to do is break down this basement membrane. So they elaborate enzymes. Remember collagen? This is full of collagen. They elaborate collagenase, an enzyme which eats away the collagen, and they can break down the basement membrane. They also are able to pull themselves along chemically. Anybody who's been sailing knows about kedging. If you have no wind, you put a bunch of sailors in a boat, you row them out with the anchor, they drop the anchor, and they pull the big boat to them. Then they row away again, drop the anchor again, and in a windless situation, the old sailing ships could kedge their way along until they got wind. That's what these cells do. These cells send chemicals out in front, they send detachment chemicals behind, and they migrate toward the chemicals and pull themselves forward, so they are actually motile. And they can move out, in this picture, through and into the tissues where they have invaded locally, and they can now grow as a group there. They can then do something else. Once they're out here, they have a way of getting into blood vessels, and they create their own blood vessels. If there's none nearby, they send out signals, chemical signals for blood vessels to grow into them. It's called tumor angiogenesis. Tumors can't grow beyond 2 to 3 millimeters without a blood supply. At 2 to 3 millimeters, they can get all the nutrients they need by diffusion, but after that, they have to have a blood supply. Judah Folkman, who was the prime investigator of this, said 30 years ago, what if we could keep all these metastases at 2 to 3 millimeters? Wouldn't hurt anybody. There'd be no physical effect of it. Why do we have to cure this disease? We don't cure diabetes, we treat it. We don't cure heart disease, we treat it. Why don't we treat cancer? And he has spent the last many decades working on discovering what those factors were that both promoted angiogenesis and inhibited them. We have natural anti-angiogenic factors, and that's a very fertile area of research now, trying to see if we can make those factors and just prevent the tumor from getting a blood supply. Very nice line of investigation. Coming back to our slide, whether we go into an angiogenic vessel or a normal vessel, these cells can also eat away the basement membrane of the vessels, whether they be capillaries, veins, arteries, or even lymphatics. They can push away the endothelial cells and make them contract, get out of the way, and then they get a free ride in the circulation, much faster than this ride, where they have to go step by step. This takes 7 seconds, and they get to a faraway organ where they reimplant and grow as a metastasis. 50 years ago, we didn't know if metastases can further metastasize. They can. So now you have exponential growth. When you go from localized tumor to metastatic disease, you are going from localized and possibly curable disease to exponential spread and almost always incurable disease. Not always, we still cure some patients in some diseases, but by and large, metastatic disease is not curable, surgically not attackable by surgery because there can be thousands, and we often can't find them. So each of these can metastasize. It doesn't always happen. Of the thousands, hundreds of thousands, and millions of cells that circulate off a primary tumor, only 0.1% will make it alive to the metastatic place, maybe even fewer. So the establishment of metastases is fought by the body. We then get basically down to 3 kinds of treatment. We can have surgery, which cuts out the mass, so we have to be able to see the mass, and it has to be in a place we can get it out without killing the patient. That's why brain tumors are difficult. Breast disease is not as difficult. We get the mass out. Where the surgeon fails is 2 places. He can't do anything about the metastases in most cases because there's too many and he can't see them. Second thing is that at the periphery of the surgical margin is a place the surgeon is likely to leave cancer cells. When I take out a tumor, I can see the gross tumor, but I can't physically see with my eyes what may be spilled cells. So I may fail right there. There's something called chemotherapy, which is an injection of a tumor toxic drug. Those circulate to the whole body. That doesn't need a target. Chemotherapy is very good for finding metastases because it'll go to every cell in the body, but it should find the metastases that we can't see. We don't have to be able to see them to treat them if we use chemotherapy. Chemotherapy deals very badly with bulk, where the surgeon does well. It does very well with very low tumor loads. So often, even when we know that a patient has widespread disease, we'll go ahead and take out the primary so that there's less of a tumor load for the chemotherapy to deal with. Finally, there's radiation. Radiation needs a target. We can't radiate the whole body. That will kill someone. You have to know what you're aiming at. But for example, in breast cancer, we can take out the lump, not remove the whole breast. The surgeon then knows that he's left tumor cells behind because if we don't radiate any of those patients, more than 30% of them will have recurrent tumor in that breast somewhere down the road. So we radiate the whole breast, and then we can reduce that recurrence rate right on down to less than 1% from 30%. So each of the modalities of treatment have their strengths and their weaknesses. Radiation needs a target, works well with few cells. Chemotherapy needs no target but can only handle a few cells. Surgery is great with bulk, doesn't do well with microscopic disease, and in the end, we often combine all three to get the best of all of them. And we cure across the board more than 50% of all patients with cancer. And in specific patients, we can get up into the high 90%. Some we do very badly with. That's the last of the things that can go wrong in our course. I hope you've enjoyed it.